Okay, this next lecture is about amino acid metabolism. And so we were going to learn about how non-essential amino acids are made by your body. And keep in mind that non-essential just simply means that we have the enzymes in order to make them, and essential means we do not have the enzymes to make those amino acids. We are going to talk about the important cofactors involved, so we'll give an introduction to those cofactors. And many of those cofactors will come up again in our single carbon donor lecture next. We will also explain some general regulation of amino acid synthesis. There are some that do have feedback inhibition, and then degradation of the amino acids can simply feed it back into the pathway from which it was originally made, or it could have distinct pathways from actual synthesis. So pretty much there are some important coenzymes. So we have pyrodoxyl phosphate, or PLP. We have already dealt with PLP in transamination reactions. And so in degradation, your PLP is going to remove the amino group. And then, of course, in synthesis, your PLP is going to donate the amino group. Tetrahydrofolate can either be FH4 or TH4. You might see either of those. And basically, tetrahydrofolate comes from folic acid, and it's going to be extremely important in moving carbons from, during the process of synthesis or degradation. And we're going to talk about many of these that involve different species of the tetrahydrofolates. And there's also tetrahydrobiopterin, and this is going to be an important cofactor for phenylalanine to tyrosine, for example. So ring hydroxylations are going to require tetrahydrobiopterin. So just a little bit about pyrodoxyl phosphate, or PLP. This has a pyridoxamine phosphate, which is when it has the amino group, the aminated form. And you're going to have, this is a cofactor, so the pyrodoxyl phosphate is going to be tightly bound to coenzymes during these transferase reactions. So there'll be tightly bound coenzymes to the enzyme and to the substrate as defined by coenzymes. Your pyrodoxyl phosphate, your PLP, is going to be bound. So here's your pyrodoxyl phosphate shown here. And it's bound to your enzyme via a lysine residue in the enzyme's active site. And so this is a non-covalent interaction. Of PLP with the lysine residue. Okay, and now folic acid. So here is a picture of folic acid. And so we have folic acid starts with folate, and we have dihydrofolate reductase, which requires NADPH. And this is going to generate dihydrofolate. And then you're going to have another dihydrofolate reductase, and this is going to make your tetrahydrofolate, which is going to be important in making more deviations of your folic acid. So some things to point out is your dihydrofolate, you have your hydrogen is going to be at position 7 and 8. So if you look at the positions here, shown here, we have a nitrogen at 5 and a nitrogen at 8, and then 6 and 7 are in between. And then your C next to your 6 is 9, and then this is going to be position 10 nitrogen. So in your dihydrofolate, this is position 8 and 7, have hydrogens. And then your tetrahydrofolate, you have those 2 hydrogens already on position 7 and 8, and you also get a hydrogen on position 5 and 6. And that's called your tetrahydrofolate. Now, tetrahydrofolate is a derivative of vitamin B9, and what happens is you're going to ingest B9 in your diet, and this goes into your intestinal cells. And then within the intestinal cells, it goes into the lysosome. And you have an enzyme in your lysosome, it's called conjugase. And this is going to remove glutamate. And this is going to release folate into the blood. And then it goes into the liver, where this is where it's stored. Okay, so the liver is the major storage of tetrahydrofolate. And so for tetrahydrofolate, you have your numbering system again. You have your 5, your 6, your 7, your 8's not really being shown here, and then a 9 and a 10. Now, your tetrahydrofolate is going to be very important in many reactions that involve methylations. These are going to be involved in gene expression, development, stability of your genome, as well as other reactions. The function of these tetrahydrofolate derivatives is to carry and transfer. So tetrahydrofolate derivatives function to carry and transfer 1 carbon units. And these are in biosynthesis reactions. Okay, and so there are various different types of tetrahydrofolates, so don't get overwhelmed. Tetrahydrofolate is that one we just talked about, has hydrogens on position 5, 6, 7, 8. That's your tetrahydrofolate. And now when you start to add carbons to this, they can only be added in certain places. So here we have a carbon that was added to the nitrogen at position 5. So this is called N5-methyl tetrahydrofolate. And then an intermediate in histidine catabolism is this N5-formimino tetrahydrofolate. So this is an intermediate. And then we have also position 10 that can pick up carbons. So your position 10 can pick up, for example, a formyl tetrahydrofolate. And then both N5 and N10 can be a bridge. So they can be a bridge for either a methylene group or a methanol group with your tetrahydrofolates. Okay, so just keep in mind, N5 is attachment site of our methyl groups, and our N5, N10 methylene tetrahydrofolate is going to be a very important cofactor for several reactions. All right, so here is just a picture that shows you all the different interconversions of your tetrahydrofolate species. I don't want you to get overwhelmed by this. We are going to talk about many of these in the coming slides, but just a few things to point out. You have at the one end your most reduced, and on the other end your most oxidized forms of tetrahydrofolate. So for what's important about your most reduced is it has an insufficient transfer potential. Okay, and so also what's important is the most common source of the 1 carbon unit is going to be that N5, N10 methylene tetrahydrofolate. Tetrahydrofolate, of course, is going to be stored form. And then depending on which reaction it's going to be involved in, it can donate or accept methyl groups. So tetrahydrofolate will accept, for example, a methyl group from serine, and this will generate glycine as well as that N5-N10 methylene tetrahydrofolate. So this is an example of a reaction, and this reaction is a serine hydroxymethyl transferase that does this. All right, so another important carbon donor is going to be SAM, or S-adenylyl methionine. SAM is going to be synthesized from methionine and ATP, and this is going to require methionine adenylyl transferase, and this generates SAM. Now, SAM is going to be the preferred cofactor for many biological reactions. And this is going to be for transfers in tissues such as your immune system. It's going to be important for cell membranes. It's going to be important for the production and breakdown of several important compounds. For example, dopamine, melatonin, and serotonin. Okay, so one of the things that's really unique about the production of SAM is that methionine has the sulfur group. So this methionine sulfur group shown here is going to attack the 5-carbon of ribose. So this requires ATP, shown here, but you're not including the phosphates. You're actually releasing all 3 phosphates. So all 3 phosphates get released, and instead you're attaching the ribose portion of ATP to methionine, and that gives you SAM. Oops, sorry, I thought I had my highlighter. So that will give you SAM. So you release 3 phosphates, and this is seen also in the synthesis of coenzyme B12. All right, so vitamin B12 has a ring. It's called a tetrapyryl ring, and it's similar to our porphyrin ring of heme. Okay, so in this case, we have a cobalt in the center, and you have many different forms, so that X can be many different groups. Our cyano group is going to be the group mainly used for vitamins and for drugs. They're going to use the cyano in that X position. Now, vitamin B12 is important in hematopoiesis. It's going to be important for neural metabolism. DNA, RNA, as well as carb, fat, and protein metabolism. So vitamin B12 is very important in our diet. There is a disease associated with vitamin B12 specifically, which is called pernicious anemia, and this is going to be very rare. It is actually an autoimmune disease, mostly caused by an autoimmune disease. where you have no intrinsic factor. And so we know that vitamin B12 has to be absorbed with intrinsic factor. And so if you don't have intrinsic factor, then you don't have vitamin B12 being absorbed. Vitamin B12 can be found mostly in eggs, milk, and poultry. So if you are a strict vegetarian, you can have nothing wrong with your intrinsic factor, but you're just not getting in your diet enough vitamin B12. And so vitamin B12 deficiencies are usually treated with excess vitamin B12, because even if you don't have intrinsic factor, you can absorb a small percentage of vitamin B12 that you ingest. So you just take in large amounts of vitamin B12. And so pernicious anemia, you're going to have large oval-shaped red blood cells, and of course less of them. So they're going to be have a variation and abnormality in both size and shape. So folate deficiency is also going to be associated with anemia, and you're going to have macro sites, which are larger than normal. It looks very similar to pernicious anemia. So you're going to have oval-shaped, large, fewer. They're going to get removed from the circulation faster than with a normal. Keep in mind that there are interconnections between folic acid and B12. And this is in the generation of SAM. And so a deficiency of either folic acid or B12 will result in reduced SAM. And so that's going to have an effect on many other processes. So you're going to have a megaloblastic anemia. And so red blood cells are going to be larger than normal. It's also referred to as macrocytic anemia. Okay, one thing to keep in mind about B12 deficiency that I should mention is B12 is stored in liver, and so it can take 3 to 5 years for a deficiency to present itself. Just simply because you have so much stored in your liver. All right, so now let's talk about the synthesis of our non-essential amino acids. So just really general, again, we're talking about non-essentials because we have the ability to synthesize them. Essentials, we don't have the enzyme to synthesize them. We are going to use amino acids, of course, for building proteins. That should be a given. So our amino acids are there to build proteins. Some other examples for amino acid usage is for the production of pyrimidine and purines. So glycine, glutamate, and aspartate all have some sort of role in either purines and pyrimidines. We also have glutamate, which is an excitatory neurotransmitter, which is important for communication in the brain in general. We have most of our non-essentials can be made from glucose. 2 non-essentials cannot because they depend on essential amino acids. And the 2 non-essential amino acids that cannot be made from glucose are tyrosine and cysteine. Those come from essential amino acids, phenylalanine and methionine, respectively. So here is an overview of our amino acid synthesis. And so just a few big takeaways from this. We have some glycolysis precursors. And then we have our TCA cycle. So what to keep in mind is that if you make glycine, serine, or cysteine, and then you break down glycine, serine, or cysteine, they usually feed back in through glycolysis. The same with our TCA cycle intermediates. If you're going to make your glutamine, glutamate, proline, arginine, aspartate, asparagine, and break them down, they usually go back into TCA cycle intermediates. Phenylalanine to tyrosine is, of course, unique because that's an essential amino acid. And then, of course, methionine here is going to provide the sulfur for cysteine. Now, the other thing to keep in mind is some of these are very simple transamination reactions, which are just shown with that TA. Others are going to have different enzymes involved in their biosynthesis and a different enzyme involved in their actual degradation or breakdown. So here is an overview. We have listed here the different amino acids, and I've color-coded them. So you have gluconeogenic that can convert to glucose. So those carbons can be used by the liver to form glucose. We have ketogenic, which are green, and those are only going to be converted to ketone bodies. And then we have blue, which are going to be converted to both. And so ketone bodies, of course, are either going to have acetyl-CoA or acetoacetate. So those would be ketogenic. You're going to have your amino acids, carbons, are going to overall become carbon dioxide, glucose precursors, ketone body precursors, or both potentially. The essential amino acids, lysine and leucine, are two strict ketogenic. So if you remember lysine, not isoleucine, lysine, here's leucine, and leucine, those are the two strict ketogenics. And then if you remember, threonine, isoleucine, tryptophan, phenylalanine, and tyrosine are ketogenic and gluconeogenic. All others, non-essential amino acids, become gluconeogenic. So just keep those in mind. Okay, let's first talk about our amino acids that are going to be derived from intermediates of glycolysis. And so if they're derived from glycolysis intermediates, when they get broken down, they're going to go back to pyruvate or glycolysis intermediates. So we basically have 4 total. We have glycine, serine, cysteine, and alanine. And alanine is pretty straightforward. You have pyrodoxyl phosphate. This is a transamination reaction. And so this is, of course, vitamin B6 is required for this. So let's first talk about glycine synthesis. So for glycine, we have 2 pathways to make glycine, from serine or threonine. And so threonine is a minor pathway, and serine to glycine is a major pathway. Okay, and so again, we're talking about glycine synthesis. So here we have glycolysis, and it goes to 3-phosphoglycerate. And this is going to generate serine. And so your serine will then be able to form glycine through serine hydroxyl methyl transferase, requires pyrodoxyl phosphate again. And in this case, your tetrahydrofolate is picking up a carbon from serine. So serine is a 3-carbon, and a glycine is a 2-carbon, right? And so your serine to glycine, you remove a carbon and transfer it to tetrahydrofolate to form that N5N10 methylene tetrahydrofolate. And then you generate glycine. Now, glycine can also be made by your threonine, and this will also generate acetaldehyde. And it's a transamination reaction with vitamin B6 again, and this will generate glycine as well. Now, glycine has a major degradative pathway. and that is through the glycine cleavage enzyme. Now, the glycine cleavage enzyme is complex. It's a complex, and so it has a P subunit, and this is the glycine dehydrogenase. And it has an H subunit, which is lipoic acid. It has a T subunit, which is a tetrahydrofolate requiring amino methyl transferase. Okay, and it has an L subunit. And the L subunit is dihydrolipoamide dehydrogenase. Now, this L subunit is the same that's in your pyruvate dehydrogenase, your alpha keto dehydrogenase, and your branch chain keto acid dehydrogenase. So this L subunit is sort of redundant across all of these different complexes. Your L subunit has the gene. It's the DLD gene. And it's on the 7q 31 to 32 position. All right, so basically what this does is you have your P subunit, which is where glycine will enter. Okay, and then from your P subunit, you're going to release one of the carbon dioxides, and this is going to be transferred to the H, lipoic acid. This will then go to the T subunit, and this is where you have your tetrahydrofolate come in. And then you're going to get your carbon released on your tetrahydrofolate. You're also going to release your nitrogen. And then you're going to regenerate, basically. And so you go back to lipoic acid, and then you transfer to your L subunit, back to your H subunit, and then regenerate back to the P subunit. And so this is how the complex works for breaking down glycine. And this is the major degraded pathway of glycine. Now there is another pathway for glycine catabolism, and this is medically important. And this is through the formation of glycoxylate as an intermediate. And so basically your glycoxylate can be oxidized to oxalate in the presence of oxygen, and this can form kidney stones. Now it doesn't have to. You can still go through a transamination reaction, and you can get alpha ketoglutarate to make an alpha hydroxy beta keto adipate, which this can be broken down into carbon dioxide and water. But the main important medical side effect of all this is the formation of kidney stones through oxalate in the presence of oxygen. All right, next up is our serine. So our serine is going to come in through our 3-phosphoglycerate. So we have, of course, our 3-phosphoglycerate dehydrogenase. So that's our dehydrogenase. And this is going to produce a 3-phosphorhydroxypyruvate. Okay, and so we're going to have a phosphoserine aminotransferase. And this uses glutamate to alpha keto. And then you get a 3-phosphoserine. And then we have a phosphoserine phosphatase. And that will lead to serine. Okay, so there is a syndrome associated with serine. It has to do with the hydroxymethyltransferase. So there are two versions of the hydroxymethyltransferase. And so the hydroxymethyltransferase is from serine to glycine. And this needs a tetrahydrofolate. And that becomes an N5N10. And so there's a cytosolic version of the hydroxymethyltransferase, and there's a mitochondria version of the hydroxymethyltransferase. And for the syndrome, it's the cytosolic version of the hydroxymethyltransferase that is going to be deficient. And so this is simply going to prevent serine from becoming glycine, and this disorder is going to lead to a lot of psychological problems, motor skill problems, growth delays, etc. Now, serine catabolism is basically you're going to have pyrodoxyl phosphate in a single reaction remove both the beta hydroxyl group and the alpha amino groups. So this is a single reaction, and it's a serine dehydratase. Okay, so that's how serine gets catabolized through dehydratase, and it can become pyruvate. Now, serine catabolism can also go to glycine. That's a form of catabolism because you're changing serine. We talked about that. Serine can also go to cysteine. We'll talk about that in just a moment. And serine can also feed back into glycolysis. So it can go to pyruvate, glycine, cysteine, glycolysis. And so in glycolysis, you have serine over here. And serine will have the ability to undergo a transamination reaction with pyruvate, which will generate alanine and will give you hydroxypyruvate. And then hydroxypyruvate can become glycerate, which then can feed back into glycolysis through 2-phosphoglycerate. Now before, if you have 3-phosphoserine, this can actually go back to the 3-phosphoglycerate. But once you form your serine is going to negatively build up. If you build up serine, it's going to negatively regulate the formation of more serine through the 3-phosphoserine, which is the phosphoserine phosphatase reaction. Now next up is cysteine. So cysteine is going to come from serine, but cysteine is going to require methionine. So the sulfur group from methionine is going to be donated and generate cysteine. Cysteine is going to require these vitamins that we've been talking about, our vitamin B12, our folate, as well as our pyrodoxyl phosphate. So cysteine synthesis is pretty involved, and it is interrelated with the formation of SAM as well. And so we have our homocysteine, which is a 4-carbon, and serine, which is our 3-carbon, and our homocysteine combined with our serine. Oops, that was bad. Hang on. There. Is going to form an intermediate called cysteine. Okay, and this is through a cysteine beta synthase. Okay, and so the cysteine beta synthase is a 7-carbon, but we want to form cysteine, which is only 3 carbons. And so we have a cysteine lyase, and this is going to form cysteine, which is our 3-carbon, and our alpha ketobutyrate. Which is our 4-carbon. And then alpha ketobutyrate can become propanyl-CoA. And you guys all know what happens with propanyl-CoA. Okay, so once you have cysteine, cysteine will negatively regulate your beta synthase. So it's a feedback regulation. Cysteine is going to be important, of course, in things like protein synthesis. Okay, but so where does methionine come into all this? Well, methionine is going to feed back into homocysteine. So we have our methionine synthase. And this is going to require an N5-methyl tetrahydrofolate. And this is going to release a tetrahydrofolate. And from this, you're going to get methionine. And methionine is a 5-carbon. So you have your 4 carbons from homocysteine plus a methyl group, and you get your 5 carbons. And you also are, so that's what you get for your methionine. And then from your methionine, with ATP, you're going to get SAM. And this is the methionine adenylyl transferase. And then your SAM, and remember in this particular transfer, it's the transfer of your ribose from the ATP and the release of the 3 phosphates to generate SAM. Now SAM is going to reconvert homocysteine and release your adenosine. Okay, and so that's pretty much what happens with your formation of cysteine. It's tightly connected to your formation of homocysteine and methionine in the production of SAM. All right, cysteine degradation. So cysteine is going to be degraded, and its degradation can go into multiple ways. So one way is shown here, where you're going to eventually form sulfide groups, and your sulfate groups can form a product called PAPS, which we'll talk about in just a moment. But let's first talk about cysteine. So cysteine dioxygenase will convert cysteine to cysteine sulfonate. Okay, and then two major things can happen here. One, you can have a decarboxylase of cysteine. And this is going to go to hypotaurine. So this should ring a bell, hopefully. And this goes to taurine, which goes to, hopefully you guys are all saying it, biosalts. Right? Now, cysteine sulfonate can also go through a transamination reaction with your pyrodoxyl phosphate, and this will give you a 3-sulfonyl pyruvate. And you can get desulfonation to give the bisulfite plus pyruvate. And that feeds back into there, and then you go on to generate urine or your PAPS formation. Okay, so let's talk a little bit about the importance of cysteine. 1, proteins. It's part of proteins like all of our essential, non-essential amino acids. It is going to be important in glutathione production. It is going to be important for taurine production. And this is for biosalts. Hopefully remember your taurocholate and your taurochenodioxycholate. Okay, and then we have the sulfur of cysteine. And that is going to be important for a few different things. This is going to be important for the formation of something called PAPS, 3-phosphoadenosine phosphosulfate. And PAPS functions as a sulfate donor in protein carb modification. And the other thing that the sulfur of cysteine does, it's a source of sulfur to form coenzyme A. Okay, so homocysteinemia, homocysteinuria, which is going to involve the defect of going from that methionine to cysteine. And this is going to lead to elevated levels of those precursor homocysteine. And these are going to mostly be caused by that beta synthase complex. And so remember, that was what brought together homocysteine serine to form cysteine, eventually, the intermediate cysteine. And so a buildup of your homocysteine and is going to lead to, or it's associated with, mental deficiencies. Our next one is alanine synthesis. All right, so alanine synthesis is simply through a transamination reaction with pyruvate. So keep in mind that alanine, remember, was very important in picking up nitrogen from tissues and bringing it to the liver. And then the liver would use the carbons of alanine for gluconeogenesis. Well, just as easily as alanine can become pyruvate, pyruvate can make alanine as well. And so that is something that is used in many tissues to pick up nitrogens or to transfer nitrogens when they're breaking down amino acids for energy during fasting. Okay, so now we're moving into TCA cycle intermediates. And so just like the glycolysis amino acids that came from glycolysis intermediates, the amino acids coming from TCA cycle intermediates will be built up by them and will be broken down back into them. So we have different sources or points, you might think, in the TCA cycle that are going to feed into amino acid synthesis. We have alpha-ketoglutarate, oxaloacetate, succinyl-CoA, and fumarate that are all going to play a role in the process of forming and degrading amino acids. So we have amino acids that are going to be related through glutamate. And so these amino acids, it's important because glutamate, of course, is an essential excitatory neurotransmitter. So it's very important in the brain. It is referred to as a major brain communicator. Okay, and so remember, histidine is an essential amino acid, so we don't make histidine, but histidine can be broken down into glutamate. All of these amino acids except for histidine are synthesized from glucose. And so the carbons from these amino acids can be reconverted back to glutamate. Glutamate is derived from glucose and can recreate glucose. So these are glutagenic amino acids. Glutamate is also important in moving around nitrogens. It's important for kidney acid-base balance. So glutamate has many roles in our body. So now let's talk a little bit more about glutamate. Glutamate can be formed from alpha-ketoglutarate. The transamination reaction, or from the glutamate dehydrogenase reaction. So we've already talked about glutamate dehydrogenase reaction, and it can go into both ways, and this requires NADPH. Sorry about that. And so it's reversible. Now, glutamine is going to have different enzymes for formation and degradation. So glutamine synthetase is going to form, glutamine, it's smaller. It's going to form glutamine from glutamate. And then when you're breaking down glutamine, you're going to use glutaminase. And glutaminase is going to release your nitrogen. This is important in the kidneys. So remember, when we're moving around nitrogens during fasting, glutamine would go to the gut and to the kidneys, and the nitrogens of glutamine is going to be important in balancing the acid secretions for metabolic acids that are released during fasting. Okay, so just keep in mind, the synthesis and the breakdown are made by two different enzymes. Now, proline is also going to come from glutamate and can be broken down into glutamate. There are different enzymes that are going to form however, the intermediates are the same. Hydroxyproline is also going to be important. We'll talk about that just briefly here. And this is going to form when proline is incorporated into collagen. And this is by a prolyl hydroxylase system. So you have proline, it requires oxygen, and our alpha keto, and then we have our procollagen, dioxygenase, and a cofactor is vitamin C. Now, one thing to keep in mind is hydroxyproline is not incorporated into proteins. However, it's 4% of all amino acids due to its placement in collagen. Okay, so proline and ornithine are going to be interlinked. And remember, ornithine is going to feed into the urea cycle. So this is all going to take place in the cytoplasm. And so you start with glutamate, and this is going to form a phosphate, and then you're going to get a ring formed and spontaneous. And this leads to a delta 1 pyroline 5 carboxylate. Okay, so this is sort of a branching point here. And so this can go on to form proline. And we have our reductase. And this requires NADPH. Okay, but also what can happen here is you can go to a glutamate gamma semialdehyde. Okay, and then this is going to lead to ornithine. It's aminotransferase. And it's a transamination reaction. Glutamate to our alpha keto again, and we form ornithine. Now, the semialdehyde just simply refers to that half of your COH groups get reduced, and the other half in your delta 1 pyro 5 carboxylate are not touched. So that's all that means. And so through glutamate, you can get proline as well as ornithine made. Now, proline degradation occurs in the mitochondria. And you have proline dehydrogenase. And this will give you a delta 1 carboxylate again. And then you have a dehydrogenase. Oops, sorry. Okay, and that can form glutamic acid. Our next amino acid is arginine. So arginine is part of the urea cycle. So remember we had arginosuccinate, and this has arginosuccinase. To get arginine and fumarate. And then remember that all of this is linked together, so ornithine to citrulline, many steps to arginine, and then back to ornithine. And so it's just a big cycle between them. And ornithine is also connected to glutamate through a transamination reaction. So we're able to go from glutamate to proline to ornithine, but also you can go from ornithine to glutamate through transamination reactions. So all of these amino acids are interconnected through glutamate, and in this case, your urea cycle as well. So degradation of arginine, again, is going to be linked to our urea cycle, as well as to glutamate and to proline. So we have arginine, and this can go from arginase to ornithine, and this will release urea, and that's part of the urea cycle. And then ornithine also can become citrulline, and that's through the carbamoyl phosphate. And then this can, through urea cycle, go back to arginine. Arginine also has a way to form citrulline through two different steps, and this is through a nitric oxide synthase. And this requires NADPH and oxygen to do this. So that's another way you can degrade arginine. And then, of course, ornithine through transamination becomes that delta pyryl carboxylate. And this can, of course, go back and forth. We can have glutamate, and we can have proline. Okay, so just depending on what the cell needs, all of these amino acids can go in all of these different directions. Of course, transamination reactions can go in reverse as well. All right, so now histidine can also give you glutamate. Histidine, of course, is an essential amino acid, and 5 histidine carbons can form the 5-carbon glutamate. This is going to require several steps. You're going to form eventually that N-formimino glutamate. This is important because this will transfer a carbon to the tetrahydrofolate pool. So you see here we have a tetrahydrofolate. You're going to pick up one of those carbons and form that N5 formimino tetrahydrofolate, which remember was an intermediate of histidine degradation. Also get glutamate from that, of course. Oops. And then your N5 formimino tetrahydrofolate will become N5N10 methynyl tetrahydrofolate, which can then become the N10 forminyl tetrahydrofolate. So these are our single carbon donor pool. Okay, depending again on what the body needs, it'll use any of those for different reactions that we'll talk about in the future lectures. All right, so we have oxaloacetate, and so we've talked many times about oxaloacetate transamination to aspartate. And so this is the major degradation of oxaloacetate, is that transamination reaction. Aspartate, of course, can then go to asparagine, and this is through asparagine synthase. And then asparagine can go back to aspartate through asparaginase. And so again, this is a case where you have two different enzymes. One is for the synthesis of asparagine, and then one is for the degradation of asparagine. Okay, so another TCA cycle intermediate is fumarate. So amino acids that form fumarate are going to be aspartate and tyrosine. So aspartate degradation, the major route is going to be that transamination back to oxaloacetate. However, a minor route for aspartate is going to be generation of fumarate in the urea cycle. So this is going to be in the cytosol. Oh, I should put minor route, because transamination to oxaloacetate would, well, it can occur in both, but minor route is okay. And so we have our fumarate in our urea cycle shown here, and how there's our aspartate coming in to help derive fumarate. And then remember, fumarate will go to malate from fumarase, and this is in the cytosol. This is also called fumarate hydratase. What's important here is defects have been linked to cancer. A glioma, which are going to be benign smooth muscle tumors. And also renal cysts are also associated with the inability of converting fumarate to malate in your cytosol. All right, the next one that can form fumarate is going to be tyrosine. So phenylalanine is converted to tyrosine, and this is through hydroxylation reaction. And this is going to require tetrahydrobiopterin. Important to point out, tetrahydrobiopterin is formed from GTP. So our cells make tetrahydrobiopterin. And the hydrogens that are important here I'm going to show are these two here. So we have two hydrogens, and these are the two hydrogens that are going to form water. They're going to be transferred to the oxygen to form water. You have another oxygen that gets picked up here, and this is going to go on to tyrosine. And then, of course, one of them goes in to form water. And then you have your quinoid dihydrobiopterin. I guess I can do that in yellow. That's formed here, and you require NADH with a dihydroopterin reductase to regenerate that tetrahydrin biopterin. And then when you regenerate tetrahydrobiopterin, it can become a cofactor for another reaction of phenylalanine to tyrosine. And tyrosine gains an alcohol group, of course, shown there. Now, the tetrahydrobiopterin is referred to as de novo synthesis because we can make it in our bodies, and it all starts with GTP. You have a GTP cyclohydroxylase. You also have a pyrovyl tetrahydrobiopterin synthase. And once it gets to this form here, it can take a couple routes to get to eventually tetrahydrobiopterin. And it requires 2 reductase steps in order to do that. Now, our tyrosine is going to be oxidized, and this oxidation is going to ultimately form fumarate, which is what our target is. It also forms acetoacetate along with fumarate, shown here. So we have our tyrosine, which is going to come from phenylalanine, and various steps can have deficiencies. So we have our PKU disease, tyrosemia 1, which is down here, tyrosemia 2, and alkaptonuria. So we're just going to quickly go through each of these. We have transient tyrosemia, and this is going to be transient because it's thought to be due to immaturity of the actual enzyme in newborns, that as the newborns develop, that enzyme becomes functional. This is mostly seen in premature newborns. That's why it's thought that it has to do with either a deficiency of the enzyme, not enough being made, or it's not as active as it will be when the infant becomes mature. And it's thought to be the hydroxyphenylpyruvate enzyme that is going to be defective. And so again, very transient. And so it's going to be treated with reducing phenylalanine in the diet until the baby matures. Tyrosemia 1 is going to lead to a cabbage-like body odor, and typically babies with tyrosemia type 1 deficiencies die within the first year of life. And tyrosemia type 1 deficiency is the enzyme that's way down at the bottom of your tyrosine and phenylalanine breakdown. And so that just means that everything upstream starts to accumulate, and over time, anything that accumulates, as we know, over time, your body has to be able to deal with it somehow, and oftentimes it becomes toxic. So tyrosemia 1 is associated with liver failure, and again, it's this hydrolase that is defective. Tyrosemia type 2 is going to be in a tyrosine aminotransferase, so that's up here. And so in this case, you get a buildup of tyrosine, and tyrosine buildup can lead to phenotypes that are presented in the skin and the eye. However, tyrosine does have other pathways it can go. It can go to melanin, for example. So there are other pathways, so it's not going to be as deleterious as more enzyme deficiencies downstream of this point. Our phenylketonuria, or PKU, is going to be this very first enzyme, so you're going to not be able to generate tyrosine. So in this case, you have a buildup of phenylalanine, which is going to, again, lead to potential damage in the brain. In this case, instead of a cabbage odor, the infants have a musty odor. If you have classic phenylketonuria, then you're going to have very severe or loss of function of your PKU. If you have a mild version, then you have some function with that hydroxylase, and it is inherited in an autosomal recessive inheritance pattern. And then also alkaptonuria, which is referred to as black urine or black bone. And alkaptonuria is going to be a defect of your oxidase, homogeneous oxidase. So homogeneous accumulates, and that is going to be oxidized, and then that is what leads to the color change of the urine and the bone. It's going to be removed, but it will accumulate, and so again, too much of something will have some effect. It can have effects downstream as you age because of the deposits. So our osteoarthritis can be one of those side effects of alkaptonuria. Okay, so now we're going to talk about our amino acids that form succinyl-CoA. And so basically we have our essential amino acids. We have the methionine, valine, isoleucine, and threonine. All are going to produce propionyl-CoA, and propionyl-CoA, of course, is what is going to make succinyl-CoA through this particular pathway. Keep in mind that we have a carboxylase that requires biotin, and we also have a mutase that requires coenzyme B12. So you still have a lot of vitamin input in your propionyl-CoA to succinyl-CoA. So methionine, which is an essential amino acid, we talked about can break down and form cysteine. So we have cysteine here via homeocysteine and cystathione. That also will generate alpha-ketobutyrate. Alpha-ketobutyrate will become propionyl-CoA. Isoleucine and valine will feed into this through propionyl-CoA immediately, and threonine will go to alpha-ketobutyrate to propionyl-CoA. Then once you have propionyl-CoA, of course, you go downstream and you get succinyl-CoA. So a few things to point out here is we have a threonine deaminase. And that's here. And this is releasing NH3, and as you know, ammonia is toxic. So ammonia in an aqueous environment will typically form the ammonium ion. Oops. And so that at a normal pH in a normal environment is what happens. So you will be able to pick up that ammonia molecule. Your alpha-ketobutyrate is going to be decarboxylated to form propionyl-CoA. So this is a decarboxylation. And keep in mind this whole process up here. So I want to sort of, let me box this area in up here. So we'll talk about this more when we talk about our single carbon donors, but this up here that's in the dotted lines is very important because up here we have a tetrahydrofolate compound, we have a vitamin B12 up here, and we have SAM, which is donating that carbon. So basically what this is showing you is the full picture of what is happening with methionine as it's going to SAM. So really you have your N5 tetrahydrofolate donating that carbon to vitamin B12. Vitamin B12 picks up that carbon and donates it to homocysteine to add it to the sulfur group of methionine. And then that sulfur group of methionine, remember, is going to, doesn't show ATP up here, but that's needed, and you get your 3 PIs released. And you form SAM. And then SAM has that carbon from tetrahydrofolate that gets donated to other processes, making other compounds or breaking them down or whatever it is. So that's what's going up at the top. And at the bottom is everything can go back into the TCA cycle through succinyl-CoA. Okay. All right, threonine is an essential amino acid, and we've talked about a few ways that threonine can be broken down. So we have that one we just discussed, which is your threonine deaminase. And this is going to release our ammonia, which is toxic, and we get alpha-ketobutyrate. And then succinyl-CoA. And then our other way we already talked about was through our threonine aldolase. And this required our transamination reaction to glycine and acetaldehyde. Right, so remember that this was a minor pathway. So this is minor, and this is more the major pathway. Okay, and finally we're at our branched chain amino acids. So branched chain amino acids make up 25% of our protein structures. So they're incorporated into our proteins. They have valine and isoleucine are going to form succinyl-CoA. So that's down here, succinyl-CoA. The two major functions of valine and isoleucine are going to be for energy generation how do I want to write this? Okay, sorry. Valine plus iso. And the other is to replenish TCA cycle intermediates. Okay, branched chain amino acids are universal fuels, and this is except for the liver. Almost every other cell in your body can metabolize branched chain amino acids, not your red blood cells. The liver does not metabolize because it's missing the branched chain alpha keto acid dehydrogenase complex. So liver does not have. Okay, so basically the first step of degradation is a transamination reaction, and that occurs for all your branched chain amino acids. And then the second step is going to be through your decarboxylation, through your alpha keto acid dehydrogenase, and that's the same. Going to have different specificities based on the different branched chain amino acid, but they all have a alpha keto acid dehydrogenase. And then you're going to generate, with valine, you're going to generate FAD, and with isoleucine, you're going to generate NADH. And this is happening in your mitochondria. Let me put that. And so, as you know, NADH will go to the electron transport chain, and FAD has several steps, but it eventually will get to the electron transport chain as well. So this will be for energy. Both of them go to the electron transport chain. And then you also are going to generate propionyl-CoA from both of these. Isoleucine can also generate acetyl-CoA. Keep that in mind. Valine cannot generate acetyl-CoA. And then both of them can also generate succinyl-CoA, so they are gluconeogenic. Isoleucine is also ketogenic. Okay? And so this is just the same. Sorry, it's a repeat. And then our third branched chain amino acid is leucine. And so leucine is going to be primarily metabolized in muscles. And leucine is ketogenic. So leucine can form acetoacetate or acetyl-CoA and become ketogenic. Leucine is not gluconeogenic. Excuse me. So big story here is the same steps. You have a transamination first. You then have your second step, which is your alpha keto acid dehydrogenase. So this is the same. Second step, and in this case, with respect to leucine, you also generate FAD, not NAD, like isoleucine. So in this respect, it's similar to valine. However, it's only ketogenic. So we have amino acids that are ketogenic, and ketogenic is going to form acetyl-CoA and acetoacetate. There are going to be 7 amino acids that are ketogenic. 5 of these, 5 are both keto plus gluco, can make glucose or ketone bodies, and 2 are strict keto. So if you remember the 2 strict keto, and then you remember the 5 that are gluco and ketogenic, then all other amino acids are glucogenic. And so the 2 that are strict ketogenics are lysine and leucine. And all the others listed here are going to be, so these are the strict ketogenics, and all the rest are going to be both. So we have our tryptophan, we have our threonine, phenylalanine, tyrosine, isoleucine. All of those 5 are going to be both keto and gluco. Now, one thing to keep in mind, I just want to make a note that lysine degradation is very complex. I'm not going to teach it here, there's no reason to. It takes 10 steps, and then you're going to eventually get to your acetyl-CoA. Okay, so let's just talk a moment about tryptophan. So our tryptophan is going to be important for a few reasons. So tryptophan, for one thing, is going to be oxidized, and this is going to become alanine, formate, and acetyl-CoA. Okay, so you have formate that gets produced, alanine gets produced, acetyl-CoA gets produced. So that's great. That's all down this whole line. Tryptophan also has a ring structure. So from its ring structure, it can form NAD. And so what this means is tryptophan can spare your diet requirement for niacin. So if you have poor niacin diets, people who eat white rice, things like that, poor niacin diet, if you supplement with tryptophan, tryptophan can form NAD and NADP from its ring structure, and you can kind of spare your body from that deficiency. Now finally, some genetic disorders. We've already talked about some of these. Phenylalanine has classic PKU, apocaptonuria. We also have tyrosine, which has tyrosemia 1 and 2. Methionine is homocystinemia. Can lead to anywhere from benign to neurologic effects. And glycine can lead to kidney stones, renal failure, etc. And so that is the end.